Hi, everyone. Thank you for joining today. My name is Alison Agneray, and I am joining you from the great state of Nevada. And I am a PhD candidate with Doctors Beth Ledger and Tom Parchman at UNR, University of Nevada in Reno. And this is one piece of my dissertation that I'll actually be presenting in February. And the title of this talk today is Taking a Community Approach to High Desert Restoration. A history of co-occurrence did not predict responses to invasion. So now, if you've ever driven through the Great Basin Desert, this is an increasingly familiar site along the side of the road. This is one location that was once native sagebrush steppe that was burned in a large wildfire, and now it is being prepared for a massive drill seeding project. And land managers now need to decide, okay, what are we going to seed here that balances the needs of ranchers and wildlife? And here in the Western US, restoration really has its roots in agriculture, where managers have selected species and populations for being nutritious for animals and also really easy to cultivate. And this sort of farming approach to ecological restoration often means planting grasses, whether they are native or not. And for years, these efforts have largely not been as successful as we would really like them to be, or they've really just resulted in non-native grass monocultures. But more recently, there's been a much greater push to include more shrubs and forbs, since the dominant native systems here are shrublands. And much of this push to restore diverse communities is driven by a more renewed focus on improving wildlife habitat for animals like sage grouse, mule deer, and lots of other animals who really rely on shrubs for nesting or forbs for insects and forage that is available year round. And all of those services aren't provided by perennial grasses alone. But still in all of these cases, we need to decide where to pull the seeds from when we're building a seed mix. And the options can quickly get complicated. Do you choose a native cultivar from a seed farm that came from a different climate, maybe from Washington, or a native, non-native species from a similar seed zone, but it's from Utah? Or, do you pick from a nearby site that is actually a different seed zone, or the same seed zone, but it's California, but you're planting all these in Nevada, So the current thinking would be that using the seeds from the same seed zone would be the best match, since you're matching climates, and it would be the best option. But more likely what people end up doing is, due to seed availability, really, seed mixes might include a little bit of all of these options. So what I want to address is, no matter which of these options that you go with, when we pick different species, we're still almost always pulling these species from completely different locations. And we never try to source all of the species for a seed mix from the same wild communities. For instance, if we're planting 5 different species, we would almost never pull all species, all 5 of those, from the same exact spot, which really made us wonder if this is a good or bad idea. There's a number of very real pros to this approach. It's cheaper, easier, more flexible to just pull seeds from wherever is available and do some shopping around. You can also pick the sort of quote unquote best seed sources for each species. But on the other hand, we might be increasing the amount of competition between native species by only choosing the so-called best sort of sources. And we could also be missing out on potential co-adaptations. What if certain sites are selecting for really phenomenal traits for restoration across multiple species? For instance, imagine a site that has burned multiple times and has really high propagule pressure from invasives. The natives that are still remaining on a site like that would have really relevant traits for surviving in a restoration context, especially if its climate matched the planting site. Further, it's really possible that interacting species from a single site have co-evolved to avoid direct competition, but they would more fully sort of use available resources, which would benefit, a restored community. Now, for this work, to ask our questions, we selected 6 different species to work on, and these are all extremely common plants that are found throughout the Great Basin. Squirreltail and sandburg bluegrass are both common restoration plants, and while Thurbers is less commonly used, it is highly palatable for wildlife and livestock. And sagebrush and rabbitbrush are ubiquitous and dominant species in the Great Basin. but sagebrush in particular does not recover naturally post-fire. And dusty maiden is another common disturbance-oriented forb that's really becoming more common to include since it does really well in disturbed areas. So we collected seeds from all 6 of these plants from 6 different communities around the Western Great Basin, where these were all growing together side by side. And we used these seeds to establish experimental communities. We planted 90 of these really enormous containers following the same planting design with 6 versions of 2 different treatments. So the first one is they either all came from the same location or they came from 6 different places sort of picked at random. And once these were all planted, each of these containers was monitored weekly for survival and annually for size. And then in their 3rd growing season, we invaded each community type with cheatgrass seeds, which is the common invasive here in the Great Basin. So with these data, we asked how community level responses differed across treatments. So we wanted to see if total productivity, which was measured as size or biomass, survival, measured as just number of deaths in a container, how many times plants need to be replaced, and then invasive suppression, so how big the cheatgrass got. Those were the community responses. And then we really expected that the single origin mixes would outperform the random mixtures by growing larger, surviving more consistently, and really suppressing the weed growth, since they could potentially use up more resources efficiently underground and leave less room for the invasive cheatgrass to grow in, either because of shared evolution in response to some site characteristic or trait character displacement, sort of niche differentiation. And then we wanted to see, in our second question, could we identify any real mechanism at work to explain differences in invasion resistance? So now let's first consider which community types were the most productive. So this figure is going to be broken into 3 panels for each of the 3 different growing seasons, with native plant volume measured in the first 2 years and then harvested total above ground biomass in the final year. So these are all going to be sorted from largest at the top down to the smallest. So here you can see that AU, which is a sympatric site all sourced from Austin, grew the largest in the first year. And all the single origin mixes will be shown as bright colors, and all of the random mixtures are going to be in gray. So the letters beside the bars are just Tukey's post hoc values and indicate significant differences. So for instance, these 3 mixtures all say A. That means that they're not considered significantly different. So now let's look at the full figure. You can start to see that there is no discernible pattern between the brightly colored sympatric communities and the gray colored random mixtures. Instead, it kind of looks like they're pretty fully intermixed, and we only found significant differences between the unique sort of community seed mixes. Like in the 3rd year, you can see we can definitively say that random mixture 2 was larger overall than many other communities. Again, here we're looking at a similar figure, but this time we're looking at mortality and then cheatgrass biomass. And again, you can see that we found no significant difference between the sympatric versus allopatric, so single origin versus random, but we did find differences across communities. Although you could say maybe if you squint a little that the random mixtures did seem to suppress cheatgrass slightly better on average, but again, that was not considered statistically significant. But you can see that again, random mixture 2, that grew to be the largest in that last figure. But here you can see it really had the fewest number of deaths and some of the smallest cheatgrass, which is interesting. So now let's look more closely at some of the more unusual communities and just consider how composition shifted over the 3 growing seasons. So here we're looking at native plant size over the years, but now each of these bars is showing the makeup of the communities. And you can see that results slightly varied by community mixture, but generally Connectus douglasii and the Alamus were generally the largest percentage of their communities in year 1. And then in the 2nd year, Poa secunda and Alamus were the largest. And then in the final year, Alamus again stayed large, but then the sagebrush, Artemisia tridentata started to grow bigger. But let's zoom in, just focus in on random mixture 2 that we keep talking about. And that was the community that grew to be the largest. and had some of the greatest survival in cheatgrass suppression. And you can see that the large result of this was almost entirely driven by the Elemis that was growing there, and that Elemis came from Alturas. And when you look at how Alturas Elemis performed with neighbors also from Alturas, which is sort of the first panel here, you can see it didn't quite have the same extraordinary performance. Another way to look at this is to consider how each population grew in communities sourced from the same location. So this is just another way of looking at this. So we're highlighting, the points in blue are showing how a plant from that location did when it was with neighbors from the same site, versus in red, that means they came from a different site. We're in a random mixture. So in Elemis from Alturas, which we were just kind of looking at, it did well in all 3 years, but it always grew better when it was next to random neighbors. And that was pretty unusual, as most species and collections didn't perform quite this differently, but it is noteworthy. And it did lead to the largest community mixture overall. So we know that most plants, really probably didn't care much about who their neighbors were, although a few did, like that notable Elemis. But let's look here at our second question, where we're seeing, can we identify any mechanism that could explain some of the differences in cheatgrass suppression, kind of shown in these figures, just showing examples of low to high cheatgrass biomass in different containers. So we found generally that larger native plants in a community overall led to smaller cheatgrass. But this relationship was not really as strong as we expected. So then we just considered, okay, were there any specific individual species that had a greater predictive effect on suppression? And we did find that the size of Elemis and Poa secunda predicted cheatgrass suppression better than overall native plant biomass and survival. And additionally, greater late season greenness, so if they stayed green for longer, that was also correlated with greater cheatgrass suppression. So the main takeaways we're left with after considering all of these results is that, community composition can occasionally affect plant performance, but simply collecting species from the same locations is actually not sufficient to produce higher ecosystem services. And our findings were really almost all unexpected. The single origin communities tended to have larger cheatgrass, which is not what we anticipated, since we thought those single origin communities would really outperform random mixers in almost every case. And now we're just wondering, okay, why did some of these randomly assembled mixes have a greater edge? For instance, some of our next steps, one thing I'm missing from this talk is just considering the environmental conditions of these sites where they came from, and then comparing them to the garden site. And likely there are some local adaptations playing a part in their performance that I didn't consider for this talk. Also, building on these results, we are performing an additional experiment to really dig into why some of these random mixes did better. So we collected from the same species again, but with this time from 16 different locations. We grew everything in the greenhouse to just quantify each population's seedling traits, and then we tested them all in the field to see which traits were tied to field success. And then we built new communities with single origin communities, random mixtures, and then also using seed mixes where we are testing these so-called best populations to see if planting seed mixes where we know their traits, if we can get better results. And this study recently wrapped up, and I'll be presenting this at my dissertation and in future talks. But you can see here, these are just a few pictures showing plants of the same age growing on the exact same day. And you can just see how visually different there were. And I can tell you there were some interesting results here. And additionally, we are currently growing out a garden to collect F1 seeds from, so that we can see if these same populations, sample them again and start running some experiments to see how maternal effects might be impacting some of these results. So if you're excited about this, keep an eye out for our future work to come. And finally, I really want to thank all of these people who made this work possible, especially my co-authors, Beth, Matt, and Tom. And I'm excited to see your questions.